Good afternoon, and many thanks to the conference and to the chairs for the invitation to ask me to give you a flavor of genomic imprinting and evolution. So there are a growing number of links between imprinted genes and human disease, and I think at this conference we've heard already several new examples. So for most of you, it will be clear what mammalian genomic imprinting is about. But nevertheless, there is a lot of confusion as to what imprinting really is. One example, in the early days, I got a letter from the police department in Beijing. And I was a bit worried because you don't like to get those letters. So I opened it, it was from the forensic department. They wanted to know about our chromatin techniques. And they wanted to have the details. So I, of course, gave them. I was very happy I was not contacted again by the police. But in the early days, there was confusion. So it's important to know which imprinting we actually talk about. So we don't talk about imprinting in relation to forensic science. We don't talk about molecular imprinting, that mechanism which makes that the shape of molecules can become altered through interactions with other molecules. We don't talk about olfactory imprinting, so the sensing of molecules, for instance, by fish who have to swim back to where they were born. This is not about behavioral imprinting, but I have to tell you, we will see that later, there are some striking similarities between parent-offspring behavioral imprinting. For instance, this imprinting which makes that young ducklings or young gooselings, like on the picture there, actually with the Conrad Lorenz, follow the first object, a moving object they see. So this is about parental genomic imprinting. So that's the marking of the parental genomes such that a number of genes become expressed from only the maternal or the paternal genome. Now, we have to be a little bit more precise because actually in different groups in different plant and animal genera, imprinting mechanisms have evolved, but very often for very difficult biological purposes. So for instance, in different insect species, genomic imprinting is the mechanism which determines sex, which determines whether these developing insect larvae will become male or female. Obviously, mammalian genomic imprinting, and actually also the mechanism of imprinting found in seed plants, is about monoallelic gene expression as a means to control dosage of gene expression. And that monoallelic gene expression in plants and animals is parent of origin dependent. So let's just have a look at these insects. I think it's curious. So that was the first mechanism of imprinting that has been described. So for instance, in Sciara flies, if you want to get a male Sciara fly, then in the early larval stage, what happens is that the paternal X chromosomes are extruded. They are removed from the cells. And that gives rise to males. So although initially males and females have the same genomes, genotypically the same, through a recognition of the paternal chromosomes in some of the larvae, there is a mechanism which makes that the paternal X chromosomes are kicked out. Very similarly, in mealybugs, male sex arises through silencing of, in this case, all the paternal chromosomes. Just to show you that, so mealybugs are these nasty insects that sit on plants in your garden and also on potatoes and different other crops. So the females are rather large and the males are very small, so phenotypically completely different. But at the early stage, from the zygote onwards and at the blastula stage, they're actually genetically identical. What happens then is in order to get the males, is that the entire paternal set of chromosomes is recognized to become inactivated. It becomes compacted, becomes heterochromatic. Whereas in the females, both the sets remain active. So this is a perfect epigenetic mechanism of genomic imprinting in insects. So let's now go to genomic imprinting in mammals. So this, as you know, is about the absolute necessity in mammals to have both a maternally-derived genome and a paternally-derived genome was discovered in about 30 years ago now, so it's actually a very recently discovered phenomenon by the group of Professor Sorani in Cambridge in the UK, by the group of Professor Salter at the time in Philadelphia, USA, but also through great efforts of Professor Katanuk in Oxford, also in the UK. Good, so there are about 120 protein-coding genes in mice and humans that have been studied a lot that are subject to genomic imprinting. Now, and it's really because of imprinting that in mammals, parthenogenetic development does is not successful. You need really both the parental genomes. So actually the group of Tomohiro Kono a while ago has shown formally that when you make a zygote which only has maternally derived genomes, but when you add these few paternally imprinted domains we know about, then you get normal development. So they made what they call a bimaternal mouse. So you don't need the paternal genome anymore, and a little bit, actually quite strikingly, a few years later they even published that these fatherless mice, they live longer than mice that have both a maternal and a paternal genome. Good, so genomic imprinting is about the need to have both the genomes, and why do you need both the genomes? Because these imprinted gene products, about 120 we know of, they actually play important roles in development, physiology, and behavior. There are many imprinted genes, about half of the ones we know about, that are specifically expressed in the brain and play important roles in behavior. We have no time to look at that in great detail. And really, another group of genes is involved in imprinted genes in the control of growth and early development. And their recent insights show that the placenta is particularly important. There are a large number of imprinted genes that are specifically imprinted in the placenta. A striking feature of mammalian imprinted genes, as opposed to imprinted genes in plants, is that they're actually clustered in the genome. They're sitting together in large domains that can make up to several megabases in size. This is the insulin-like growth factor 2 domain here to the right. So this gene, IGF-2, is expressed from the paternal chromosome, and it is actually flanked by another imprinted gene which plays also a role in the control of fetal growth. Now, this monoallelic expression of those genes is controlled by a differentially methylated region, which conveniently we call an imprinting control region. And this imprinting control region itself, and that's the important message for today, is controlled by allele-specific DNA methylation. So at this ICR, that methylation imprint is found on the maternal chromosome in all the somatic cells, both in boys and girls, there's no difference between the sexes, and it originates from the sperm. Now, right next door, there is another domain. I think we've had several presentations on this already. There is this KCNQ1 gene we just heard about. So it is the KCNQ1 imprinted domain, which is controlled by another of these ICRs, these differentially methylated regions. This ICR is methylated on the maternal chromosome in the embryo, and after birth, that methylation is maintained. And this methylation originates from the oocyte. Now, in this domain, there are a number of genes that are specifically expressed on the maternal chromosome and not on the paternal. And the key gene involved in growth is a negative regulator of the cell cycle, the CDK1C gene. Good, so imprinting is actually pretty easy. This is about regulatory CpG islands, CpG dinucleotide-rich sequences that have regulatory functions that, however, function only on one of the two copies, because they are marked by germline-derived monoallelic DNA methylation. So one allele has DNA methylation and the other one not. And it's really this which is at the heart of the evolution of genomic imprinting, and we'll see that later. So when did imprinting arise? Well, it is actually a very young phenomenon. It evolved about 170, 180 million years ago during the Jurassic period. So imprinted genes are found in marsupials, eutherians, like in humans and mice, but they're not found in monotremes, like in the platypus, and there's no evidence for parental genomic imprinting in salamanders and chickens, for instance. So the group of Marilyn Renfree and Kaneko Ishino, Renfree in Australia and Kaneko Ishino in Japan, have actually traced back the evolution of these regulatory CPG islands, these imprinting control regions. And that really coincides very, very well with the acquisition of imprinted gene expression, as far as we know that. So here's just one of the images from very nice work done by these two groups, which show that the CPG richness of these regions is conserved in the species where there is imprinted gene expression. And actually these regions, the sequences also are relatively well conserved between groups of animals that show imprinted gene expression, but not with groups of animals that show no imprinted gene expression. So evolution of imprinting is about the evolution of these highly specialized CPG islands. Of course, we need to explain, one needs to explain why imprinting evolved, because it's not necessarily very healthy to only have one of the two parental copies which is active. If you lose the expression on that one remaining copy, then you lose expression altogether. So many theories have been proposed to explain the evolution of imprinting, and each of these explains the imprinted gene expression patterns of a number of imprinted genes, but not all. And we will see later that there are a number of confounding factors related to these theories, and one of these is that actually imprinted genes are intricately linked to each other in their regulation and in their functions. So there has been extensive co-evolution of patterns of imprinted gene expression, and it will become important to look at groups of genes rather than individual genes. So what do these theories all say? That you have one copy of the gene coming from mother, the matry gene, one copy coming from mother, patry gene, and there is some non-equivalence of the matrygenic allele and the patrygenic allele with respect to fitness. And then selection over time is going to favor the imprinted silencing of one of the two alleles. So one very attractive hypothesis proposed about 10 years ago by Wolf and Hager is the maternal-offspring co-adaptation theory. It says that there needs to be a match between the alleles expressed by the mother and alleles that become expressed in the developing young one. So there is a correlation between the matry genes, of course, and the maternal genotype in the offspring. And through epistatic selection and interaction effects, there is selection of the expression of the matry gene so that there is the perfect match with the genotype of the mother. So this is really about interaction between gene products between the mother and the developing offspring. And this is more about the nature of the product than about the level of expression. Another much older theory, which is based on the kinship theory, has been proposed by David Haig and Westoby in 1989, also involving Chris Graham a few years later. That's really based on the observation that in social groups, and particularly in offspring, for instance, of individual females, there is much sharing of matry genes, but much less so of patry genes. So obviously a female produces during her life offspring that have all her genotype, but will have different paternal genotypes, because in mammals are polygamous. So there is a higher relatedness of the matry genes than the patry genes in offspring, but also in social groups. That makes that certain patry genes can make profit of that. For instance, by making the offspring grow bigger. In that particular case, that patry gene will be transmitted more readily because that offspring will be healthier, stronger, and better. So this then gives a battle between, if you wish, the interest of the maternal genome and that of the paternal genome, such that for some genes, the matry gene will be expressed and the patry gene will become silenced over time through gene level selection. And for other genes, the metry gene will be silenced and the peto gene will be expressed. Now, this is all very complicated, and I find it always very complicated to explain this, but there is some sort of a popular derivation of this theory, which says this is the parent-offspring conflict theory. And we have to be very careful, because the way this theory is described is remarkably similar to behavioral imprinting, parent-offspring imprinting. So remember the IGF2 gene. In mice and humans, it's expressed from the paternal genome and it stimulates growth. So you could see this as a paternal genome which wants to achieve growth in the offspring and therefore be transmitted more readily. So in this popular view, the father instructs the developing baby, grow big and strong, use all the maternal resources. Future babies may not have my alleles. The father is very selfish. The mother gets worried. She's going to instruct the developing baby and say, grow well, but leave some resources for me and my future babies, because she knows that she will have further pregnancies and there will be more children born, and she can't give all her resources. So conversely, at least in mice and polymorphically in humans, there is this IGF2 receptor gene which is imprinted, which is a negative regulator of growth. So the father says to the IGF2 receptor gene, you have to be switched off, so my offspring will grow bigger. And the mother says the opposite. Now, this is all very well, but you have to realize that there is never an instance that parents are actually instructing a developing baby. The imprints I talked about, these methylation marks, are established in the developing germ cells. So there's well before individuals are adult and before they are parents. Good. So genomic imprinting is also the evolution of mechanisms. So what's new? Well, this is not really new. So we know since 10, 15 years, the enzymes involved in the establishment of imprints in sperm and in the oocyte. Those, of course, these imprints are then maintained in the zygote. But really what is remarkable is that these allelic methylation marks are maintained throughout development. They are somatically maintained. So this is controlled by DNMT1. And really this is remarkable. Genome-wide studies by different groups in Europe and the States have shown during the last years that there are many regions that become marked in oocytes and sperm, many regulatory regions. But the only ones, probably the only ones, which retain their differential DNA methylation status during the critical early stages of development are those imprinting control regions. So we now know that the specificity of genomic imprinting is determined by the somatic maintenance during very early development, much more so than their establishment in the germ cells. Many things become methylated in germ cells, but what counts is whether you remain methylated in the early embryo. So ICRs are somatically fully stable. One allele will always remain methylated in the early embryo, and the other always unmethylated. Well, always, not always, because very rarely, and that is now we go to the human diseases, these imprinting disorders, very rarely that maintenance process is perturbed. So for instance, in Silver-Russell syndrome, which I think doesn't need introduction to this audience, this intrauterine growth restriction syndrome, which is mostly sporadic, there is a perturbation of imprinting. So here are, I think this is a monozygotic twin sisters, and this girl here was very small at birth and will always remain small. So what happens in this case, so this was discovered by the groups of Machine and Lebouc, professor here in Paris, and by others. In this syndrome, in the majority of cases, what happens is that the imprint that controls the expression of the monoallelic expression of IGF2 is actually lost. And this is thought to happen early in development. I would like to point out that there has been a very interesting consensus statement meeting, and the first publication was out very recently. So have a look at this. This tries to bring a system into description of Silver-Russell syndrome and its epigenetic and genetic defects. Good, so what is important relative to genomic imprinting and the evolution of imprinting is to understand which mechanisms maintain these marks during the early embryogenesis. And I can't go through all the details. I would just like to summarize a lot of work from our lab and other labs is that there is a highly specialized chromatin structure at these ICRs, at the DNA methylated allele, which involves histone methylation, alternative histones, for instance, histone H3, and the recruitment of non-histone proteins. Now we have to assume, it's difficult to prove this in models, that this particular specialized structure ensures that the methylation is maintained efficiently. Conversely, on the unmethylated allele, there is consistently a type of active chromatin which prevents DNA methylation. And I will leave it here. A very interesting insight, and from the last few years on from different imprinting disorders, is that the loss of DNA methylation at those ICRs often occurs in concert at multiple imprinted loci. This really presumes that there may be common underlying factors that are perturbed, and maybe even genetically perturbed. Another important insight, which I would like to share with you, is that there are all the time more examples of how imprinted genes and imprinted gene products are linked. So for instance, and these are just a few examples, if you're interested, there's an interesting article, interesting review written by Manus Patton together with us on this trans-regulatory links. An interesting example is this imprinted growth factor involved in transient neonatal diabetes, which controls many other imprinted loci. So one imprinted factor controls many other imprinted regions. Then, of course, you know, if you look at the products, then, for instance, in the IGF-2 insulin signaling pathway, there are multiple components that are subject to genomic imprinting. What my group and others find very interesting is that most imprinted gene products are not proteins. They're actually non-coding RNAs. So there are hundreds of imprinted non-coding RNAs, and we only just start to look at the function of those, but we are already seeing that many of these imprinted non-coding RNAs control the imprinted expression of protein coding imprinted genes. So there you see very complex links, not always in cis, but also in trans. There are now examples of imprinted loci having an effect on other imprinted loci. This evolved, but it also has consequences for the way we have to think about genomic imprinting and why it evolved and how it works. So we can't go a lot more into this. I would just like at the end to go back to this cartoon so that in mammals, because of genomic imprinting, you need both a maternal and a paternal genome. And that is because, of course, because of these protein coding genes we know about, for which it is absolutely essential that we have both the genomes in order to get expression of all of them. And they play many important roles in placental development, fetal growth, physiology, and behavior. But I would like to point out, for what is new in research, is that we know of hundreds of regulatory non-coding RNAs that are controlled by imprinting as well. Just to give you an idea, in humans and mice, about 7 to 8% of all the microRNAs are imprinted. They're controlled by genomic imprinting. And I think tomorrow Jérôme Cavalier will give you one very nice example of a cluster of imprinted genes and how that imprinted expression is important in that particular case for the postnatal adaptation to the change in food intake. During the last 20 years, most studies have been performed on embryos and on adult animals, and I think we've forgotten a little bit about the important role imprinted genes play, of course, in nutrient transfer, and we have actually forgotten a little bit about the importance in placental development and function. But again, here, things are going very fast. There are all the time more examples that link the levels of the placental levels of expression of imprinted genes to, for instance, birth rate. So one example is this factor I already mentioned to you, this ZEC1 PLEGL1 imprinted transcription factor, which has been studied a lot by the group of Laurent Junot in Montpellier. And it has been found by the group, I think, of David Monk in Barcelona, that levels of PLEGL1 are downregulated in intrauterine growth restriction placentae. Interestingly, PLEGL1 is now known to actually specifically regulate this imprinted transcription factor, specifically regulates the IGF-2 H19 locus. It actually controls the main enhancer at that imprinted locus. So that's another example of one imprinted gene controlling another. So I think we're going to have quite some interesting examples of this kind. Now, what I find most striking, and this is where I will stop my talk very shortly, is that genome-wide studies in human placentae, mostly by the groups of Robinson in Canada and the Kelsey group in Cambridge in the UK, but also a group of David Monk in Barcelona, have shown during the last 2 to 3 years that there are actually hundreds, literally hundreds, of new placenta-specific maternally methylated, differentially methylated regions. So those are CpG islands that in most cases become methylated in the oocyte. That methylation is maintained in the early embryo and in the extra-embryonic lineages, but that very same methylation is lost in the embryonic lineages. So most of these ICRs, they become unmethylated in somatic tissues. And for instance, here's one. This is a new CpG island, which is methylated in the oocyte. Here you see that methylation patch. It's fully unmethylated in sperm, so it is an imprinted DMR. If you look in the blastocyst, you get the expected 50% of methylation, one chromosome methylated, the other not. And also in placentae, there is 50% methylation. But look, in the somatic tissues, this methylation is lost. Interestingly, most of these new placenta-specific DMRs, maybe imprinting control regions, we don't know yet, seem to be primate-specific. So this is a very recent and possibly fast evolution of imprinting, specifically in the placenta, the tissue where imprinted gene expression is supposed to be very important. And the other interesting thing is, maybe because this is an ongoing process of evolution, but the group of Robinson in Canada has shown actually this year, and there's unpublished work by the Monk group, that many of these recently evolved or evolving potential imprinting control regions actually show rather variable levels of methylation between different placentae. And some of them actually show polymorphic imprinting. So isn't that interesting? We are capturing here the evolution of imprinting right here. So I think we're going to leave it there, and I would like to thank my team for allowing me to go to this meeting. And I would like to point out this COST network, UCID, the European Network of Congenital Imprinting Disorders. For anybody interested in genomic imprinting disorders, there is a fabulous website, and you can get much more detailed information than I provided to you on imprinting disorders and the epigenetic and genetic deregulation that is causing those disorders. And here I will leave it, and I thank you very much.